mentor to me. So Dr. Giorgio graduated from Vienna with a PhD in neurobiology, and he has been a member of the Life Sciences Department since 2010. He's going to speak to us about sleep in fruit flies and whether it is vital or not. Great. So let's start.
Right. Thank you, Giza, for the introduction. Thanks, everyone here. Yes, as Giza said, I'm trying to show you some of the data we've been working on. I'll try to address what I think is a rather important question about sleep, and that is whether we should consider sleep as a vital necessity or not. What do I mean by vital necessity? It'll become clearer throughout the talk, of course, what I mean by vital necessity, but a good similarity that might help understanding this is whether sleep is as vital as food. Okay, so there is no doubt, I think, that sleep has some vital implication. If you don't sleep, you're sleep deprived, and you drive a car, you're probably going to kill yourself and the people around you. But it doesn't mean that it is the same vitality that you get from calories in food. We know that all animals need to eat, and they all need to eat a certain amount just to sustain a basic vital activity within energy. And so the question is does the same apply to sleep? And this, is obviously quite an important question, but is surprisingly not really well addressed in the literature. And so we try to give our contribution using Drosophila as a model system. The reason why we can do this is because we accept this postulate, if you wish, arbitrarily accept that sleep is an evolutionary conserved phenomenon. And characteristics that look like sleep have been found in, we say, virtually every animal that we studied so far, from as simple as jellyfish, is an animal without a brain, all the way to mammals and insects, and exotic animals as elephants, for instance. And so this no animals has been able to escape this slavery of sleep in a way. So this evolutionary conservation is quite staggering. But at the same time, it shows itself with a great amount of variety. And so you have in the animal kingdom animals that sleep as little as 2 hours a day, like giraffes, 3 hours a day, like elephants, and animals that sleep as much as 20 hours a day, 21 hours a day, like the bat or the koala. And so you have to, whatever hypothesis of sleep function you try to put forward has to be able to come to terms with this variety. And you have to explain why some animals need 20 hours and other animals need 3 hours of sleep. Even more puzzling, there are animals that can change actually sleep amount depending on ecological situations. So birds are a fantastic example. Birds in normal condition might require only 7 or 8 hours of sleep. But at a certain point during their life, birds can cope with basically virtually no sleep. And so it's the case of migratory birds. They migrate from South America, for instance, to Alaska. They fly through the ocean. During the time, as far as we know, they never sleep. That's a trip that takes weeks to be accomplished. And as they reach their destination in Alaska, they actually, some of them are able to forgo sleep for further 2 or 3 months, just to dedicate 24/7 all of their time to mating and to courtship. And so these are animals that therefore can change dramatically their sleep requirement depending on the ecological situation. And again, if you want to explain what sleep does, what is the function of sleep, you have to come to terms with this flexibility. Right, so why do we study sleep in Drosophila? Well, there are many reasons to use Drosophila as an animal model, as you probably are familiar with. The most important is that we believe that flies sleep, otherwise we wouldn't be studying this on the first place. We believe that flies sleep because what we call sleep satisfies this 5 textbook definition of behavioral correlates of sleep. So sleep in flies is associated with is a period of quiescence associated with a species-specific posture. So this animal change posture during sleep. There is an increase in arousal threshold, meaning that in normal condition it would just be enough to shadow your hand over the eye of the flies to start them up and away. But when they are awake, when they are asleep, you actually need a stronger stimulus like a bang or a noise. This period of inactivity is quickly reversible to wakefulness, which is what distinguishes sleep from coma, and is homeostatically regulated, meaning that if you sleep deprive an animal, they'll be more tired the day after. And finally, there is an interaction with the clock, meaning that the sleep tends to happen always at the same time of the day, which in the case of flies is relatively similar to ours in terms of the fact that they sleep mostly at night and they are mostly awake at sunset and sunrise, as most insects are. You also get some bonuses like conserved signaling pathway, conserved function of neurotransmitters, and conserved effect of drugs. So if you give hypnotic drugs to flies, they fall asleep. If you give stimulant flies, they stay awake. So that tells you that the conservation is not just at the behavioral level, but there is also a evolutionary conservation at the molecular level. And so that is really a very solid ground to assume that anything you find in Drosophila will then be able to translate to other species. Right. Before we dive into the data, it's important that I explain you how we analyze sleep in Drosophila, and I do this also because it's we actually use a system that was instrumental to generate our data, and that system is slightly different from what everybody else uses. So the most commonly used system to analyze sleep in Drosophila actually relies on activity measurements. So you put the flies in a tube, like this one. The tube is about 7 centimeters long, so it will host only one animal. There's food on one end, cotton on the other, and all the animal can do during their time in the tube is moving back and forth. As they move back and forth and walk throughout the length of the tube, they'll cross an infrared beam, which is invisible to them. And this infrared beam will therefore detect activity and send the signal to the computer. In a way, if the computer knows that the fly is walking, obviously it can assume that they are not sleeping. And on the other end, if they are not walking, if they are not crossing the beam, after a while you can start counting sleep. As you probably already have sorted out yourself, this system is simple but not necessarily precise. If the animal is not walking throughout the distance of the tube, if it's not crossing the infrared beam, then no activity will be perceived and sleep might be overestimated. And this might happen, for instance, every time the animal is feeding. By the food, spending possibly also hours at once. No activity might be detected for hours, and sleep might be overestimated by a large amount. So to overcome this limitation, we actually created a system that is initially custom-made. It's now published, and it's based on two off-the-shelf electronic components. The core of this, the heart of this is a Raspberry Pi. Raspberry Pi is a microcomputer you might be familiar with. It's a machine that was originally developed by University of Cambridge. It's a real computer. You can connect a keyboard to it, a monitor, a mouse, and do all your Facebook, Gmail, all your Word and writing activities. But it's very inexpensive. The price of a Raspberry Pi is about 15 to 20 pounds. And we also use an Arduino, which is something similar to a Raspberry Pi, but also only for microprocessing power. So you can actually build robots with it in quite an easy way. So when we put these two things together, we create a machine that, as I said, we call the ethoscope. What is an ethoscope? Is a machine about this size that hosts the Raspberry Pi on the top case. You see it here. And attached to the Raspberry Pi, there is a camera. Shown here in green. The camera is pointing towards the bottom of the machine where you'll put your flies. And so what happens is that the computer will look at the flies at every single time. So you're not relying on any infrared beam anymore, but you're simply just looking at what the flies are doing with a resolution of about 5 times a second. So this is as I say, it was published a couple of years ago. We are keen in other people using it. It's taking, obviously, quite a bit of attention within the sleep community. But in principle, you can use it to do much more than just sleep analysis. In the paper, we provide instructions on how to build an ethoscope, and you can do it the way we do it, so that it's using 3D printing technologies. Or if you are a bit more creative, you can actually play around building a paperscope that is cutting a pattern and fold it together. Or if you are up for a nice weekend, you can follow instruction and build a Lego scope. So this is actually one of the supplementary information of the paper. We had to remove the Lego logo, but we managed to sneak in the Lego minifigure in this. Right, so what can you do? As I say, we use it mainly for sleep, but in principle you can look at any behavior. That's what the name etoscope represents. The behavior of choice will depend on the arena that you build. The arena is physically the place in which you put the flies and in which the flies move and live. And so for sleep analysis, we normally use this arena that hosts the same tubes that I showed you before. For other studies, we have an arena that hosts longer tubes. You can play around with having something a bit more complex if you want to study learning and memory, for instance, spatial memory, and so on. You then control everything from the network. The etoscope talks to each other via Wi-Fi, and you can collect data into the cloud and analyzing from your computer. Right, so what kind of behavior are we looking at? What kind of behavior are we talking about? One important peculiarity of this system is not just simply looking at where the animal is, but is also doing some online analysis of what the animals are doing. And for doing this, it uses an algorithm of statistical machine learning. So basically, we annotated several hundred videos. We feed the machine learning algorithm with this annotation, and so the machine learning will understand and we be able to discriminate a fly that is walking from a fly that is immobile or performing what we call micro movements. So here are some examples. This is a fly that is actually walking up and for the tube. This is a fly that is immobile, so possibly asleep. And this is a fly that is feeding, sitting by the food. And finally, a fly that is grooming. So these micro movements are not, you should not think of those as small twitches of the body, but rather like in-place motion. Things like clearly signs of wakefulness, like feeding or grooming. And yet, nevertheless, they are very important to be detected because the other system could not even remotely look at those. And what kind of data you get? This is an example of raw data. This is data of 5 female flies and 5 male flies. Obviously, you see them here over the period of 5 days. And if we zoom in this yellow window, we now look at better resolution over the period of 4 hours. What you have here is the position of the animal on the y-axis over time on the x-axis. And obviously, if the animal is moving and walking a lot, you will get this zigzagging pattern back and forth. This is an animal that is actually moving and walking. If they stay immobile, then you'll see a steady horizontal line, like in this case. So this animal here is immobile for half an hour or almost an hour, which is then counted as immobility and sleep. You'll see animals, like in this case here, that are yet still immobile, but they are immobile by the food, and they actually perform some motion, some movements while they are there, and that is a sign that they're actually not sleeping, but rather feeding, spending the time by the food. And so this is the kind of better estimation of sleep amount that you get with the system that you would not be getting before. So if you compare then the analysis, the sleep analysis done with etoscope with a sleep analysis that is done with a commercial system, the infrared beam system, you get something like this. On the left panel you have males, on the right panel you have females, and etoscope sleep will be indicated by a continuous line, while the infrared beam sleep by a dashed line. And you see that in terms of shape, so in terms of quality of the data, it's not dramatically different, right? The shape is kind of the same. The animals tend to sleep at night, mostly. Males also have this daily sleep, which we call a siesta. Females also tend to sleep at night, and they have less of a siesta, whatever system you look, compared to males. But even though the shape of the profile is similar, you already realize that the quantitative data are very different. And in fact, if you go and quantify the difference, you see that it's quite staggering. With the infrared beam split, you would estimate that male flies sleep as much as 80–85% of the time, while in fact they sleep 50% of the time. And for females, you would estimate about 60% of the time spent asleep, while in fact it's more like 25% of the time. So there is almost a threefold difference between the two systems, which is even more clear in females. So now this is important, but it was less surprising. Our lab and other laboratories had shown, obviously, the limitation of the other system, had shown that video tracking was definitely much better at estimating sleep amount. But what was really surprising then is that because Ethoscope allow you to do high throughput analysis, we have about 100 of those machines in the lab now, and each machine has a throughput of about 20 flies, so we can, in principle, screen 2,000 flies a day. So because we can do finally high throughput analysis with this kind of resolution, then we can uncover differences in a population that is actually much larger than what is normally studied. And so that's what we did. We analyzed sleep in almost 900 virgin females and almost 500 males. And this is the bulk of the data. What you have in the upper panel in pink and blue, stereotypical colors, is a sleep amount of each single individual. So each line will be a single different animal. And you see that in males, across a population of 500 siblings, you have variability that goes from 60% of the time spent asleep in the most long sleeping cases to about 20% of the time spent asleep. In females, you go from something like 40% to a tail that almost reaches 0. And this is where it was really surprising. It was the point where we found a decent number of flies that slept only a few minutes a day. We have about 5% of the flies that sleep less than half an hour a day. And the most extreme cases are flies that sleep 4 minutes a day. So when I say at the beginning that no animals that shows no sign of sleep has ever been found, I wasn't lying. It's really that kind of surprise that we had in front of us. Right, so we obviously, this was, as I say, unexpected. So we wanted to make sure we weren't, the Ethoscope will not play a trick on us. So what we did is we recorded videos for some of the animals. We looked at the videos with our own eyes, annotated this data. We also look at the single tracing for each animal. And if you look, for instance, at this one, this is one of the short sleeping flies, female flies. The roll tracing looks like this. You have lots of walking activity during the light phase of the day. And then during the night phase of the day, you still have walking activity. But most of this walking activity is actually relegated to one side of the chamber. So if you draw the virtual infrared beam across the midline of the tube, you'll see that a lot of walking actually happens without touching that infrared beam. So that explains the difference, the quantitative difference that we see in our system that no one could observe before. You will also see that all of this activity is actually on the bottom part of the tube, which is the one where the food is. So it confirms that females have this very strong food-related behavior that can be confused by using the wrong system, can be confused with sleep. We can do also some spatial analysis, temporal and spatial analysis of the type of behaviors we detect. So that's what we do here, for instance. We compare male and female flies, and we look at where the walking position can be detected. And when you do this, you find that walking can be detected pretty much along the length of the tube, on average, across the 24 hours, which is what you would expect, because a lot of the walking is up and down. Quiescence, on the other hand, seems to be very biased towards a very specific position in the tube, which is here, about 1 centimeter away from the food. And that's the same in males and females. And finally, micro-movements, which are things like grooming and feeding, are found to happen in yet another position. So in females, the majority of micro-movements actually happen in close proximity of the food. And so that shows that flies have a specific position for sleep and a specific position for micro-movements, and it kind of addresses the question on whether they could do the same two things at the same time. It's unlikely that they do that. So in short, what we're doing is we're moving from a bidimensional analysis in which flies can either be walking or not walking, or in fact crossing or not crossing the midline of the tube, to a tridimensional definition where you have walking, quiescence, and micro-movement. So that means that at any given time, say about 30 minutes throughout the day, you can score a single animal depending on their behavioral profile. So we can say this animal now has spent 20% of the time walking, 20% of the time quiescence, and 60% of the time with micromovements. And if you do this throughout the 24 hours for all the animals, you get then an idea of how they behave as a group. And it should be here. It's not. Let's see if I can show it to you. I should have used my laptop. There we go. No? Okay. So this is what I was meant to show you. On the upper part, you have female flies, all of the female flies in the first data set. In the bottom part, you have the male flies, and each dot is a single animal. And as they move in time, they move into this virtual space. And what you see is that they move basically as a swarm, right? So they all tend to do the same thing at the same time. So that is the first indication that a lot of their behavior is intrinsic. These flies, obviously, they don't live together. They are each one in a separate tube, but they tend to do the same thing at the same time, depending on mainly the time of the day. So that's really a clear sign of circadian behavior. And you see that females spend a lot of the time between walking and micromoving. And there is only one point during the day, which is here, by the end of the night, where they have an increase in quiescence. And so most of the sleep that we see in females happens in about ZT20, ZT22, so the last 2 to 4 hours of the night, while micromovements happen during the day, but also throughout the rest of the night. Okay. So when you then flatten this data from a movie to a 2D figure, it looks like something like this. This is the whole population averaged together, and you see that the pattern that you observe with male flies over the 24 hours is very different from the pattern that you observe with female flies over the 24 hours. So that is what we call behavioral fingerprinting, and it gives you an idea of how, by looking not just simply a sleep amount, but a behavioral profile, you can separate population of individuals. So we wanted to then look at behavioral fingerprinting and see whether we could use it to explain more about our phenotype. And we did this experiment in which we analyzed how mating changes sleep in flies. Why did we do that? I need to do a short digression here to explain, first of all, why there is such a staggering difference in sleep amount between female flies and male flies. And this is down to basically metabolic necessity, and that comes down to the dietary conditions. So females lay eggs, and they mate once in their life, female flies, and then they tend to fertilize inside their abdomen embryos and lay fertilized embryos as eggs. And they can lay about 200 eggs a day. This each egg is in theory an adult, right? So it's an embryo that will have to develop into a full-formed fly. And so it needs a lot of energy to go through all phases of development. So it means that each egg is a really protein-rich, energy-rich concentrate. So in order to lay so many eggs, females need to sustain a very demanding diet, which is mainly a protein-rich diet. And this is not just Drosophila, it's pretty much every insect. And it's also the reason why, if you are out and about in southern Europe, it was always the female mosquitoes that are going to bite you. It's never going to be the male mosquitoes. So female mosquitoes bite you because they want your blood, because they want the proteins in your blood. The male mosquitoes, they don't care about you. They are totally fine with sugar that they found in fruits and in the wild. And so this different necessity of energy conditions create basically a very different lifestyle between female flies and male flies. And the lifestyle changes even more after mating, as you can imagine, because all this process of laying eggs, fertilized embryos, starts after sexual mating. And so what we did is we took female flies, we put them in our tubes, as I said before, and we let them there for rest for 3 or 4 days, and then at one point we introduce a male in the same tube, and that's what we call the mating event. The male stays there for about a couple of hours and courts the females and eventually mates. Now, a tube like the one I showed you before is not the most romantic setting you can think of, and that means that the mating event is actually not 100% successful. It's conveniently, in fact, only 50% successful. So we end up with an experiment in which all the females experience the same behavioral, they have the same behavioral experience, so they're all courted by the male, but only 50% of them will actually mate. And so that is convenient because it gives you a very good control to experimental animals condition. So then when you remove the males, you can then look at how mating has changed sleep in those populations. And that's what we have here. In green is the mated flies, and in gray is the unmated control, which, as I say, they just experience exactly the same treatment, but beside the mating event. And what you see is that sleep is heavily reduced in mated flies. It goes down pretty much immediately. And especially this is seen the first night, the second night, the third night, and probably farther on. You also see that the position of the average position of the animal in the tube, which is here on the lower graph, changes as a consequence, and they spend much more time by the food after mating than they will do otherwise compared to their unmated control. And so that tells you that the mating event will reduce even further sleep in this animal, will shift the sleep amount even farther to the right. And so the question is, perhaps this explains the short sleep phenotype that we've seen to start with. Maybe those female flies that we found sleep only a few minutes a day, they were either accidentally mated throughout the process, or maybe they thought they were mated. Something in their brain that clicked this post-mating behavior, and that's why they behave in this way. And so we did behavioral profiling to compare the three populations and to see whether that's the case. What we're comparing here is on the gray triangle, you have the non-mated control, so the flies that have not successfully mated despite the encounter with a male. In green, you have the mated flies, so the ones that actually went through the whole process. And in pink, you also have flies that were not mated, but whose sleep is identical in terms of amount to the green population. Okay, so the green population that reduces sleep a lot has the same sleep amount as the pink population, which is way less than the gray population. And yet what you see here is that the shape is already indicatively different, and it tells you already something. It tells you that the shape of the non-mated females is actually much more similar than the one that the mated females. The mated females have a different behavioral profile. And then if you cluster this and try to find proximities, you'll find indeed that non-mated flies, they cluster together, and mated flies are farther apart. So it doesn't matter that the green and the pink population have the same sleep amount. The overall behavior is different and is more divided. You can actually use it for dividing non-mated versus mated animals. So that tells you that no, the flies that we found that were short sleeping had nothing to do with mating in the first place. And it tells you that scoring behavior in such a way can give you a vision that is much more powerful than simply say, how long do the flies sleep? Right, so we having found these flies that can cope with so little sleep, the question then becomes, is this a peculiarity of only these animals, these lucky or unlucky animals, depending how you see it, that can sleep so little, or any fly can cope with a few minutes of sleep a day? And so the way to test this would be to do sleep deprivation. Sleep deprivation is a system that is used basically by every sleep scientist, whether they're working with men, with rodents, with flies. And it's a tricky process because as you might imagine, it always carries a certain amount of confounding factors. It would be most commonly stress. So some people don't really control for stress thoroughly when they do sleep deprivation. So you have condition, for instance, in, let's see. So you have condition like this one, in which, in this case, rats are placed on a treadmill, and as you see, they are very eager to move and to run. And they actually don't really do this for fun, but they do this because if they stop moving, the treadmill will push them in this area of the machine where they get a little electric shock. And that is painful enough to give them motivation to run as long as the machine is running. So obviously, as you can imagine, this is not really the most controlled system to perform sleep deprivation. With flies, most people will use another system that is basically a bit more rudimental, but decently effective. You put flies on an orbital shaker, the same orbital shaker you find on the lab where you put your Western blots. And when you do this, basically this is what the flies experience. This is the condition, and then the shaker turns on, and is a bit of this Santorini earthquake every minute or so. There is not electric shock, possibly not much pain involved with this, but it's definitely a treatment that is quite invasive. And most of all, it will not differentiate depending on the flies. So basically, all the flies get the same stimulus at the same moment. Whether they are sleeping or not, they get the disturbance. Probably conceptually the best system to do sleep deprivation in animal models is this one. It's known as the Discover Water system. And it was developed by Rex Schaffen in the 60s and 70s. The way it works is you put two animals on the same platform. It's a disc that rotates on water. These animals cannot see each other because they are physically in two separated rooms. There is a wall in between. So they cannot see each other, but they are sitting on the same platform. And one animal will be the experimental animal, the other animal will be the control animal. They are both hooked to a machine that reads their EEG, and so the machine will know whether the animal is falling asleep or not. And as soon as the experimental animal falls asleep, the disc will start rotating. The rotation will push the animal towards the wall, so the animal has only one decision to make. Either wake up and walk against the direction of the disc, or be pushed into the water. And rats really hate water, so they really, again, have a very good incentive not to do that. What is the advantage of this system? The advantage of this system is that you have a control animal on the same platform that gets exactly the very same stimuli that the experimental animal gets. But the control animal can sleep whenever the experimental animal is awake. So the extent of sleep deprivation is very different. The experimental animal can get, we can aim at least at getting 100% sleep deprivation efficiency, but with the control animal, you are more like into 40-50% territory. And so when you do this experiment with rodents, we know that they die eventually, after 21 days on average. Rex Schaffen basically spent his career trying to understand why they died. He looked at every organ, from the brain to the lungs to the liver, anything you can imagine, never really found anything. So at the moment, we still don't know why these animals die after sleep deprivation. But we know that when they do die, they look really miserable, they lose fur, their metabolism change, their body temperature increases, they eat more, but they lose weight. So they definitely have a systemic metabolic condition that probably is linked to death. So we developed something conceptually similar for our flies. The etoscope have the ability to record activity in real time, as I said. So that means that in real time, that can feed feedback to a machine. And so we have machines, for instance, that give flies odors that we can use to send stimuli. We also have machines that give them light, mainly for optogenetics experiments. But we also have machines that can rotate tubes. And so the idea is that now you have a fly into the tube, and as the fly stops moving, the motor will rotate, and the tube will rotate as a consequence. And it looks like this. So focus on this fly here, the first fly on the left. It's been moving for a while. And then so that's the kind of stimulus we do. Okay, so it's a quick, fast rotation, 200 RPMs for about 1 or 2 seconds. All right, so then having built this machine, the question is, can we sleep deprive the flies with it? And the first experiment we did was an analysis of how the flies respond in general to sleep deprivation. We focused on short-term sleep deprivation. So what we did is we kept flies in the tube for habituation for a few days, and then at the beginning of this night 0 if you wish, we started sleep deprivation protocol. So for 12 hours, the flies experienced tube rotations. And the tube rotations were triggered by a certain amount of immobility. So you can set a trigger in time. You can say, rotate the tube if the fly doesn't move for 20 seconds, or rotate the tube if the fly doesn't move for 40 seconds, and so on, as for activity trigger of the length of your choice. And what we found is we tried numerous triggers. We found 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different triggers from 20 seconds to 1,000 seconds. And when you do this, obviously, you see that the efficiency of sleep deprivation is proportional to the length of inversely proportional to the length of the trigger. So if the trigger is very short, obviously the flies will pretty much lose all of their sleep. If the trigger is very long, the flies will only lose little of their sleep. But in all cases, whether the trigger was short or whether the trigger was long, flies show what we call a sleep rebound. So that means that when you stop the experiment, the flies want to recover the sleep they've lost. They sleep longer immediately after you stop the experiment. This is something that is part of the homeostatic control of sleep, which I talked in the beginning. It's crucial of the definition of sleep in every animal. It's something that we all experience. If you stay awake all night or part of the night, the day after you will want to sleep longer. This is common probably in everyone in this room when it comes to Saturday and Sunday, for instance, unless you have small young children, you tend to sleep longer on Saturday and Sunday morning, and that's what we call the social jet lag. It's a way to basically recover from sleep lost during the week. And flies show this rebound, but interestingly, they show this rebound in any case. So whether you disturb them with a 20-second interval or 1,000-second intervals, you still see a rebound. And why is this surprising? It's surprising because flies' sleep is not as consolidated as human sleep. They're not animals that go to bed in the evening and then wake up in the morning. They have, say, numerous sleep episodes throughout the night. And the number of sleep episodes, which are actually longer than 1,000 seconds, which is about more than 15 minutes, is not very high. So that means that in the last most extreme sleep deprivation condition, we only rotate the tube of the animal on average 4 times a night. Okay, so this animal are disturbed about 4 times a night. They experience only 4 tube rotations a night, and yet the day after they show a rebound. And that doesn't really make too much sense in terms of sleep lost to sleep recovered. But it's something, again, that is, if you look carefully, is common to many animal species, even humans. If you have, again, I use the kids metaphor because I experienced it myself, but if you have children who wake you up in the night even just once because they want to be fed or whatever, that's enough to feel sleep deprived in the morning. And so that we know that consolidated sleep has a value by itself, but again, we have no idea why this is so. It might just be simply that being disturbed during sleep induces a response, a punishment if you wish, that says you shall not be disturbed while you sleep. Please don't do this again. Right, so the next big question is then the one that tries to, goes back to the first couple of slides. Is sleep deprivation lethal? So can we kill the flies by just removing sleep from them? And so to look at this, we asked this question, but we kind of expected an answer from it because a similar experiment was already done. It was published actually in this paper, which is a paper on Nature that dates back to 2002. And what they do in this paper is described here. So they say individual cantonés, which are basically non-mutant flies, were kept awake for 70 hours by tapping on their tubes when they stopped moving. And so basically they had an undergraduate student possibly sitting there in front of those flies, spending 70 hours tapping on the tube whenever he thought the flies were stopping to move. And this was done on 12 individual flies. And out of 12 flies, 2 of them died after 60 hours of continuous wakefulness, whereas 2 more died by 67, 70 hours. So in principle, when you read that sleep deprivation is lethal in all animals, the fly experiment is this one. It's not a very good experiment, as you can see. It's very limited in numbers. The methodology is really, primitive, if you wish. But it was 20 years ago almost, so this was the tools that people thought it was appropriate to use. So we were expecting then to see lethality in the range of 60, 70 hours. When we did a similar experiment in male and females, we let the flies in their tubes for a few days, and then we start the sleep deprivation protocol. And as you see, the sleep deprivation is quite efficient, both in males and females. And you would expect them to die somewhere here. And instead, the sleep deprivation went on, and all the flies basically concluded the experiment with the control. And if you look at the longevity in these animals, in males, we found no statistical difference in length of life between the control and the sleep-deprived animals. And in females, we found only a very minor difference of about 3 days between control and sleep-deprived animals. So it tells you that even removing most of the sleep in these animals has not had an effect on their ability to survive. And only a minor effect, in fact, if any, in females. So that's, it goes together with the first set of results, right, that I showed you before. We found, on one end, we found flies that sleep very little. On the other end, we found that any fly can sleep very little. So it tells you, concluding that there might not be a vital function for sleep in our setup. One thing that we are now interested in looking at, and we basically create the next chapter of the study that we will work on in the future, is then investigating what happens in terms of sleep pressure in these animals. All right, if they, apparently don't need sleep in this sense, why do they feel tired with sleep deprivation? So, studying sleep pressure and studying sleep rebound is the direction in which we're going next. And what are we seeing so far? So we found interesting already insights by conducting this experiment. This is an experiment in which flies are sleep deprived not for 12 hours, but not even for life or something in between. In fact, it's sleep deprived for 10 days. Okay, so the treatment is the same. 20 seconds of immobility will trigger the tube rotation, and this treatment will last for about 10 days. So you can study how sleep pressure builds within the range of 10 days, and also you can study as the experiment, as the sleep deprivation ends, you can study how they respond to it. And that is what kind of rebound they have. So if you remove 10 days worth of sleep, will they sleep for 10 days following that? We don't expect that. No animal does that, including humans. But the question is still valid. So we looked at that. We deprived, as I say, male and female flies for, in this case, 9.5 days. And then we stopped the deprivation and we look at the rebound. And what you find is this. You found that the rebound in males and in females is present, but is nothing to write home about. It's not a major rebound. It's a rebound that is in line with 12 hours of sleep deprivation. Again, this is similar to what was observed with other animals, including humans. There is only one case of a young American student who went on awake for about 11 days. His intention was to enter the Guinness Book of Record. Name was Randy Gardner. And so Randy was awake for 10 days. During these 10 days, he was okay. He was spending his time with friends and doctors who were overlooking his performance. He was playing basketball. He was playing chess. He had episodes of irritability and some aggression, but otherwise, for most of the day, he was okay. And then as the sleep deprivation ended, he went to sleep. He slept a few hours more, and that was it. That was the end of it. And the similar thing we see now in flies. So we see a rebound, but it's nothing major. Interestingly, in females, we actually see a rebound that manifests itself during the first morning and possibly during the first night, and also the subsequent day and night. So it's actually, yes, it's not a very strong rebound, but it's more long-lasting. And how does sleep pressure builds up in these animals? Well, what we did is we look at the number of tube rotations over time, and the number of tube rotation is a proxy of sleep pressure, if you think about it. Because if we rotate the tube, and the fly immediately responds, wakes up, and starts walking, then we are not going to rotate the tube again until they start moving. But if we rotate the tube, they just move a little bit and then fall asleep again, then they will experience another tube rotation in 20 seconds, and then another in 20 seconds, right? And so the more frequent the rotation, the more tired the flies are, because they are less responsive. And so when you do this, you see that the number of rotation actually is not steady, is not going steadily throughout the period of the experiment, but it goes up and down, up and down, up and down, and follows a circadian pattern. So this is average over the 24 hours. Number of rotation is here, and you see that there's hardly any rotation at this point of the day, and more during the night. So what does it mean? As I told you before, flies tend to be crepuscular animals. They are more active at sunset and sunrise. Most insects are like this because it's a good strategy to avoid the hot hours of the day. These animals, they are going to be active at sunset and sunrise no matter what. It doesn't matter that they've been sleep deprived for 10 days. When sunset comes and the internal clock says you should be active, they are active. We actually hardly need any rotation of the tube to keep them awake. And so that tells you that the circadian clock is still regulating the internal pressure even after many days of sleep deprivation. In a way, this is known and it's probably experienced by most of you. Every time you have jet lag, you feel tired, no matter how much sleep you had the night before. That's irrelevant. You know, if the time comes, you will be tired anyway. But at the same time, you feel active in the same way when it's time to be active. So I think it really shines as a different light on the strength of circadian rhythm in regulating our sleep pressure. We know that it's involved, but I think it's much more involved than what we think it is. And we can do experiment now in a circadian mutant, like in this case. This is a mutant in the clock gene, kept in constant darkness. It's a condition where the circadian clock is completely gone and non-functional. And when you do this, you see that the animal basically does not show any pattern of activity in terms of sleep pressure. So they obviously, they're always tired the same way throughout the 24 hours. And you can also calculate how this sleep pressure grows with time. And so when you do this, also in this condition, you find that the sleep deprivation by itself contributes to only something like 11% of the build-up of sleep pressure. And most of the sleep pressure is actually explained by the circadian rhythm. 70% of sleep pressure is explained by the circadian rhythm. So again, it tells you that circadian rhythm is really important in determining this. Also, so what I will show you, that we build a system to detect activity in flies using it's called etoscope. By using this system, we then found wild-type, non-mutant flies that are virtually sleepless, and we show that we can sleep-deprive animals for basically all their life without a consequence on their longevity. What does it mean? I anticipated in the beginning that we were trying to see whether sleep has any similitude with food in terms of function, right? I think I'll stick with this analogy. If you think about food, there are 3 types of calories you can ingest. There are these vital calories that you absolutely need to survive. It's about 2,000 calories in an adult person. If you don't eat those, you basically starve to death. Even if you spend all your day watching Netflix on the sofa, you really need those calories to just function. And then there is a number of calories which are useful. So if you're training, if you're working, if you're studying, you need extra energy to actually perform at that level. But finally, there is a number of calories that is neither vital nor useful. And we all experience those to a different extent. Those are the calories you get when you get the extra piece of pie, the extra beer. I propose that the same is true for sleep, that there is an amount of sleep that is possibly vital. Now, with our results, we cannot exclude vital sleep because we still give some sleep to flies. Remember that we rotate the tube after 20 seconds. So it's possible that many of those 20 seconds interval are actually 20 seconds interval of sleep. We can definitely conclude that uninterrupted sleep is not a vital necessity, but we cannot conclude that all sleep is not a vital necessity. And then there is sleep clearly that is useful, right? There is a lot of literature about the effect of sleep on memory, on performance, on whatever, reflexes, cognitive abilities, but also immune system. And so that is useful function of sleep. And finally, there is an amount of sleep that is probably neither useful nor vital. And that amount, which going on a limb, I would say is most of it, accounts, for instance, for the difference between the sleep in the bat and sleep in the elephant. Why one sleeps 4 hours and the other 20 hours? And so, the question is because they can. The bat can afford to stay in his cave 20 hours a day, escape from predation, escape from danger, escape from weather. The elephant cannot. The elephant has to feed itself continuously because of the mole and because of the vegetarian diet. And so one important message of this similarity is that if you try to remove sleep from a bat, even just half an hour of sleep from a bat, they'll probably be sleep deprived. In the same way as if you try to remove half an hour or 1 hour of sleep from you guys, you'll also be sleep deprived. But the same is true if you try to remove unnecessary calories, okay? That's what being in a diet means. If you go on a diet, you try to remove your unnecessary calories. And your body will not like that. Your body will trick you into thinking that those calories you're trying to get rid of are actually vital, right? Try and renounce to a pie, your body will tell you, no, that's something if you don't eat that pie, you're gonna die, right? That's the kind of message you get from your stomach. And so you should not, you cannot trust your pressure to do something, whether it's sleep or feeding, necessarily to judge whether that something is absolutely vital or not. All right, so that was my last slide. Most of this work, well, all of this work, in fact, was conducted by Quentin Geisman and Esteban Beckwith. Quentin used to be a PhD student in the lab. He now moved for his postdoc to Canada. And Esteban was a postdoc, and he's still a postdoc, but he moved on to a new adventure now. That's it. Thanks a lot.
Giorgio, for speaking to us.
Oh, wonderful. Thank you.
A little of appreciation. Is there any question from, actually, yeah.
So with this model, you score it testing in vital comparisons, right? So what about animals where you have, like, as I'm not moving in vital survival, like sleeping is vital for not being infected with predation and stuff?
Right.
How do you kind of separate sleep in terms of it has some vital function for survival of the organism and
Right. So
not being killed by something?
I guess it comes down to what in the definition of vital. As I said before, sleep can be vital in the sense that if you are sleep deprived and you drive a car, you're going to kill yourself. So that is vital in that sense. But that is not the same vital meaning that you give to those 2,000 calories a day you have to get. So it's that's the difference. So if an animal needs to stay immobile, not to be prey of a predator, it's still vital, but it's vital in a different way, right? It's more like driving the car.
Professor Wilson.
Can you rule out local sleep in the brains of your researchers?
Oh no, absolutely, we cannot rule out local sleep. They probably have micro sleep, right? So this 20 seconds of sleep that they surely experience throughout the day. That's a good definition of micro sleep, and they might have as well a local sleep. Yes, clearly, we remove their normal. We interfere and completely remove their normal sleep pattern. So whether they compensate with something very artificious, we don't know what is possible.
Dr. Ricardo?
You have short sleeping flies, long sleeping flies. Are they always short sleepers? Always long sleepers? The same fly?
Yeah. So we look at that actually. What we've done is we put flies in the tube and we measure their sleep for a week. And then we remove the flies from the tube and we put it in another tube, a new tube with new food, new cotton, in a new endoscope, in a new incubator in another side of the room. So everything about their life changes. But what you find when you do this is that the sleep amount remains the same. So there is almost a one-to-one correlation inherently. So it's something intrinsic to the animal. We don't know what it is. It's possibly something partly genetics, but genetics definitely cannot explain all of it because all these animals are siblings. So they are not isogenic, so there are some small differences in the genome, but they are siblings, so they're very close. And so genetics cannot definitely explain. So it's probably some stochastic developmental feature.
Yeah, I just have a quick question. So when we sleep deprive an animal, we always try to replace it with something. So for flies for female flies, for example, that naturally sleep only, for example, 10 minutes, what do they do during their time? Do they just eat?
Well, they live more. I mean, they experience the world more. So it's possible that, they, for instance, in principle, they might feed more, they might groom more. There's not much more they can do in this environment. But yes, we didn't look at whether, for instance, they get more food during that time. Likely not, because maybe they have a different food hygiene, if you wish. But yeah, the short answer, we don't know whether they compensate during their wakeful time with feeding or other things like this. One crucial thing is that in our system, they don't have to necessarily walk very long distances in order to stay awake, right? So they can actually just move a little bit, or even just perform in place motion. So you don't have a strong component of fatigue. You know, they're not running on treadmill. So fatigue is probably not a component of the system.
All right. If there's no more questions, I'd like to thank you all for coming again.